everyone. As promised, here's the little video of all those first objectives of that first lecture. It was very heavy, right? There was a lot of those objectives. A lot of this is review. You've seen it in Gen Chem or in other different chem classes, right? So this is really a refresher, and we kind of glazed through a lot of it last class and didn't really go in depth. So as I said, I figured I'd make this video just to really go in depth on each topic and to make sure if ever there's something that's you're uncertain of, you can watch it, try to understand it, and then from there, if you have questions, then you can come see me because we kind of skimmed through it very quickly. So this will help to go through the basics. As I said, it's not most of this won't be tested on directly. It'll be integrated into what you need to know, right? The things that I think are a bit more important, I'll definitely point out. But yeah, we'll go from there and we'll see what things will be a bit more important for exams. As I said, here's my email. You have my office hours, where my office is. You can reach me anytime, okay? And I'll be teaching Unit 1. And then around the beginning of Unit 2, Chris should be back to take a look at these last 2 units with you. As I said, anonymous feedback form. If ever there's anything that you want to suggest to me, something that you're struggling with, and you would like me to change in the way I'm teaching, go ahead. Please go fill out this form, and it's anonymous, right? So you can give me some feedback without needing to hesitate about anything. I really do appreciate some feedback, okay? All right, here's our to-do list for today. So for lecture 1, the things I would like you to do is first, well, you're watching this video, so that's a good sign. And you can follow along with your PowerPoint, right? And you can pause whenever you need to, so it'll be helpful. Take notes on any questions and either bring them to class Thursday, come to my office hours, right? Or book an appointment, whatever. Send me a MIO or set up a Zoom meeting with me, okay? Anything works. I'm very flexible and accessible, okay? So that's for the video. Now, a lot of people like reading instead and learn better that way. So Professor Chris made an amazing set of notes, right? You can find them on Leia. And if you look at pages 1 through 7, that's all the pages you'll need for this topic. So if you want to skim through them, that would be a good idea to kind of get a different take on things, maybe. Also, it's a good pre-lecture thing. So if you want to read them before coming to lecture, I'd also suggest you that. At the end of this lecture, I gave you the pages that would be good for next lecture to read. So you choose what's best for you, okay? But this page 1 to 7 is good for the chemistry of life section. Do as many questions from the lecture 1 question package. So I'll be uploading that soon. So really, I'll give you a lot of questions to do, right? And it'll be good reviewed. Some concepts might be a bit trickier, but that's good. I want to challenge you, and maybe there'll be some questions that are a bit more ambiguous, right? And you'll need to think about it or come up with like some kind of rationale to some questions. So I'll let you guys play with those, and again, you can come to my office hours and see me anytime to look at those. Okay, lecture 1, chemistry of life. Why chemistry? This is a biology course. Well, it's because living organisms are chemical machines, right? So atoms come together and create bonds together, composing molecules. And then those new molecules are also continually undergoing chemical reactions to become new molecules. This may be from hydrolysis, molecules might be breaking apart, or synthesis reactions. Maybe sometimes molecules are synthesizing together, right? Synthesis reactions can bring molecules together into big specialized groups, and that we call macromolecules. Macro means big, right? And molecule, well, it's a molecule, right? So big molecules. This includes very, very important molecules we will be talking about this year. These you should be super familiar with throughout the year. You'll be experts on all four of these fields, okay? Carbohydrates, right? Those sugars, right? Lipids, right? Those fats. Proteins are everywhere, and nucleic acids, right? Your genetic material is made out of these nucleic acids. Now, I asked you in class, right, to name me a type of carbohydrate, a lipid, a protein, and nucleic acid, right? And I gave you the example of starch for carbohydrates, triglycerides, triglycerol for lipids, right? Different enzymes, lactase, amylase for protein, and nucleic acid, DNA. Someone said mRNA, someone said tRNA, right? All of those were super good. They're types of nucleic acids. So these macromolecules are the building blocks and make up organelles, like the mitochondria, right? This contains lipids, carbohydrates, proteins, and nucleic acids, right? So let's think about what exactly, is where you might find these things in different organelles, right? So I gave you this example over here, right, where you'll find a lot of carbohydrates, right? You have these long, in the chloroplast, right? You have these long polymers, these starches, right, that you'll find in the chloroplast. Nucleic acids, you have your chromosomes that are made up of this DNA, right? These filaments, which are protein kind of cytoarchitecture of your cell, they kind of keep the cell all held together in the matrix, right? These are made of a bunch of these amino acids that are all kind of linked together in a chain and form this big filament, okay? So that's something that's very important. And then fat, if you think of triglycerides, right, you'll find them in these fat cells, in these adipose cells, right? So all that to say that then these organelles create a cell which acts like a small city, right? The nucleus is like the parliament, making decisions, passing laws. You go out, the mRNA, oh, let's make this gene, let's make that one, right? The nucleus is the big decision maker. You have the cell membrane, right? This is like border control. Oh, you come in. Nope, not you. I gotta let you in. There's like a little checkpoint over there at the cell membrane, right? The mitochondria is the energy power plant, right? So actually using water, so I'd call it hydro electricity. But the mitochondria is kind of creating all this energy for the cell so that I could use it and giving it away to certain parts of the cell to use. So then these cells come together and form tissues, right? Let's say cardiac tissue, muscle tissue, and then those tissues form organs, right? And then the organs form your instructor or you, right? So all this from small atomic particles all the way up to a human, right? There's different levels of organization, and it's that very intricate organization that makes us function the way we are. And that's what biology is all about, and that's what this course will be all about. Understanding more the smaller things that are going on in the body, the microscopic things, to see what could be explaining the way our body functions. Okay, so this big list of objectives, right, that we need to get through. So the first one is define matter, elements, atoms, molecules, and compounds, right? So first, the basics, atoms, right? So let's talk about matter first. Anything that takes up space and has mass is called matter, right? All organisms are made up of matter. Atoms on the other hand, are the basic units that all matter is made of, right? It's that simplest unit. They are made up of subatomic particles, right? So you have protons that are positively charged and reside in the nucleus of the atom. Neutrons have no charge and weigh approximately the same as protons, and they are also the other component of the nucleus. And then the nucleus is surrounded by a cloud of electrons. Yes, they orbit the nucleus, but not in those elliptical paths that are shown in textbooks. Even this one is not super accurate, the path of the electrons, right? They are kept in orbit around the nucleus because there's an electromagnetic field of attraction between the positive charge of the protons and the negative charge of the electrons, right? Now it's more of an orbital, right? It's an electron cloud. The electrons have a negative charge, right? So that's how they're being attracted to the nucleus. As the protons in the nucleus have this positive charge, right? So that's not too bad. Now let's take a closer look at electrons. So the energy of an electron in the shells is called potential energy. This is the energy required to move an electron from the nucleus to the shell, right? The shells furthest from the nucleus have the greatest potential energy, okay? So if energy is absorbed by an atom, one or more electrons may be boosted to a higher level, right? To a higher energy level. If the absorbed energy is later released and lost by the atom, the electrons fall back to the lower energy shell, and the released energy is emitted as heat, this thermal energy, and or vibration or light, right? This concept is really important when it comes to understanding cell resp and photosynthesis. Consider what happens to a car, right, when it's exposed to sunlight. Sunlight excites the electrons on the surface of the car to a higher energy level. When the electrons fall back down to their original energy level, the car's surface heats up, right? This thermal energy can be transferred to the air or your hand if it touches the car, right? So it's important to keep in mind when we're considering bond formation and hydrolysis, breaking apart things, and we're looking at the process of cell resp and photosynthesis. Why is it that you're getting this energy or releasing it, right? It's all due to these electrons jumping around in the shell levels, right? Okay, so elements now. Not atoms, elements. So elements, we're going to say, so usually the number of protons, electrons, and neutrons, right, in an atom are different depending on the element you're looking at, right? So because of this, each atom has a unique name, a unique mass, and size. The different types of atoms are called elements, right? So for example, the atom that makes up the element oxygen has 8 protons, 8 neutrons, and 8 electrons, right? That's set in stone. While the atom that makes up the element we call sodium has 11 protons, 12 neutrons, and 11 electrons, right? So all this to say that they both have very different chemistries, right? Now, differentiating molecules and compounds, right? A molecule is formed when 2 or more atoms are bonded together. So for example, H2, O2, H2O, and methane over here, right? all of these are molecules, right? You have a molecule of dihydrogen, a molecule of water, a molecule of dioxygen, a molecule of methane. If you had oxygen alone sitting there, then it would not be considered a molecule, right? It's just an atom in that case. Now, for compounds, compounds are a bit different. Compounds are formed, the definition is when two or more different atoms, that's the key here, different atoms are bonded together, okay? So, dihydrogen is two of the same elements, right? You have hydrogen and hydrogen, so that's not a compound. A compound needs to compound things. It needs to take different atoms and bring them together. So look at water. Water is a compound. It brings together hydrogen, oxygen, right? O2, oxygen, right, is not a compound because it brings together two oxygens. Now, what about methane over here? Methane brings carbon, hydrogen. So yeah, we have more than one element. We're perfectly fine, right? So I said this in class, but it takes a minute to digest this. All compounds are molecules, but not all molecules are compounds, right? So they're just different definitions. Okay, next thing. Differentiate between atomic number and atomic mass. So, atomic mass versus the atomic number, right? Normally, when we do not have isotopes, so when we have a normal atom, the number of protons is equal to the number of electrons when everything's normal, right? Now, the mass number equals the number of protons plus the number of neutrons, right? So, for carbon, you have 6 protons, 6 neutrons. The atomic number is just the number of protons, right? Now, the atomic mass equals the mean weight of protons, neutrons, electrons of an element, including all of its constituent isotopes. So, basically, an element's atomic mass is the sum of the weight of protons, neutrons, and electrons, but of depending how many isotopes we have, right? Look here. The mass is not exactly 12, as it would be 6 plus 6, right? Because this number takes into account the mass and relative abundance of isotopes that have more neutrons than normal, right? So it's a little bit higher than 12 because carbon 13, carbon 14, they have extra neutrons that we need to account for. They're very rare. They're not as abundant, right, in nature. So it's a relative average of how abundant they are, and so it doesn't really make a big difference to the carbon 12 mass. It's just a little bit higher. Now, we are next going to understand what is meant by the term isotope, right? We keep saying isotope. An isotope, right, has different mass numbers than its original element, right? So this is when atoms of the same element have a different number of these neutrons, right? In this case, right, normal carbon, right, is 6 protons and 6 neutrons, and that makes a mass of 12, right? That makes a mass of exactly 12, right? And its abundance in nature is actually 99%, right? That's crazy. Now, carbon 13 is a little different, okay? Carbon 13 has this extra neutron, right? So it's going to be, its mass is going to be 13, right? 6 plus 7. And this is about 1% of the time, right? And carbon 14 is even rarer, okay? So that's what I was saying when I was saying the mass is average depending on its abundance, right? So that carbon 13 is going to account for a little bit of the weight, right? The average weight. Now, okay, let's talk about stability of these isotopes, right? Stable isotopes are ones that exist and do not decay. Carbon-12 and carbon-13 are both very stable isotopes, okay? Radioactive ones that decay over time, they are releasing energy and changing their atomic structure, okay? So they're a bit different. The nucleus of those decay spontaneously and give off these particles of energy radiation, right? Now, let's talk about how it's used in medicine. Okay, so stable isotopes can be tracked due to their different weight and can determine where atoms are going in biological processes. But also in environmental sciences, we can kind of track where, let's say carbon, right? Like carbon cycles, you can look at these isotopes and see where the carbon is going in the cycle and study it a bit more. Now, radioactive isotopes, right? These are the ones that decay over time and can be naturally eliminated, right? They're able to decay. So we use radioactive tracers, radiotherapy for cancer patients. This is using radioactive cobalt, actually. And radioactive glucose during PET scans. It's actually called this fluorodeoxyglucose, FDG, right? Here, you'll see this. I put a picture of a PET scan of this 68-year-old woman with invasive lobular cancer, metastatic to the liver, okay? So you'll see that in the top panel, okay? It's pointing to this little dot, right? And you see the PET scan. But what happened was there's this radioactive glucose, and wherever there's high metabolism of the cells, you'll see this later on, these cancer cells are metabolizing very fast. They're growing very quickly and expanding, right? So there would be an anomaly in the PET scan. But then you see after systemic therapy in the bottom panel, right, you see that little dot is gone, right? So that's a really good application in medicine. These carbon isotopes, radioactive isotopes, are also used for carbon dating because they have a fixed half-life, right? So when they decay, we know exactly how much time it'll take for them to decay, right? And so we can kind of figure out the timing by working backwards. Now, state the elements that make up 96% of living matter. Okay, so there are over 100 elements that have been discovered or produced and these are very important for living organisms that rely heavily on only a small fraction of these. Actually, only 25 elements are essential for life. Carbon, hydrogen, nitrogen, and oxygen account for 96% of this. They call them the big four here in the little table, okay? That counts for 96. Then that's just 4 elements, right? These must be important. Phosphorus and sulfur are the next important ones, okay? Followed by calcium, potassium, sodium, chloride, magnesium, okay? So these are the other ones that are important. And the remainder of them are called trace elements and are needed in small quantities such as iron and iodine. Okay, so here let's start with those top 4 elements: oxygen, carbon, hydrogen, nitrogen. Oxygen, right, is the percent body mass, including water, H2O, right? 65% of our body has its mass is made up from this oxygen. That's crazy, right? It's a component of water, and it's very important for cellular respiration, okay? This whole, you'll see the water and the electron transport chain. So it's very important. Now, carbon, right, forms the backbone of all organic molecules, right? So that's very important, and it's at like 19%, right? Hydrogen is a component of water, so it's super important. It's only 9.3, but think of it. This is not a very heavy atom, so that's why it might not be much of your percent body mass. And it's a component of many organic molecules, okay? Then nitrogen is very important for proteins. Actually, amino acids, an amino group is actually with nitrogen in it, okay? So, and also nucleic acids. We're going to see in DNA and RNA that there's these nitrogens very often in the structures. The 5 next major elements are found in the human body are calcium, phosphorus. So phosphorus is important to us because it's present in the backbone of nucleic acids. We will see that. And it's very important for energy transfer, ATP, right? And signaling within the cell. Potassium, right? Sulfur. Sulfur is small, but it's really essential for proteins also. And chloride, okay? Calcium, potassium, and chloride are very important in the nervous system for synaptic talking between the synapses of neurons, okay? So that we will maybe take a look at a bit later on. Now, okay, those trace elements, right? So I don't want you to go memorize these, but just know these are required by an organism in very small quantities, less than 0.01% of mass, right? We need it though. Let me show you an example. Iodine, right, is an essential ingredient of a hormone produced by the thyroid gland, right? Here, thyroxine, you need this in your thyroid, right? Insufficient iodine in the diet can result in things going wrong with your thyroid gland. It's called a goiter. If your thyroid gland starts swelling up, that's a goiter, and there's problems going on. It's trying to overproduce, right? Now, something really cool is that radioactive isotopes are used to treat thyroid cancer, okay? So here we have a liquid with some iodine that has radioactive isotopes. So the iodine will be absorbed by your gut, and then it'll go directly to the thyroid gland, and it'll attack the thyroid gland and reduce all this big goiter, right? So this is it'll basically kill the thyroid, right? So this is interesting because it's very targeted. Iodine is not used, from my knowledge, anywhere else in the body necessarily. It's really just for this one purpose of making this thyroxine, okay? Let's look at iron. This is another of those trace elements, okay? So it's a very important component of oxygen-carrying proteins in the body, which is hemoglobin. So here's our red blood cells on the right, erythrocytes, right? We see one red blood cell. If you look into the red blood cell, we find hemoglobin. Hemoglobin is what carries oxygen from the lungs all the way to the tissue, right? It goes through your circulation, brings it to any tissue that needs it, right? It's also what brings back the CO2. So there's this transport in and out and in and out. But how does the oxygen just bind there? Well, hemoglobin is made up of 4 hemes, okay? If we look at one heme in the center there, you see this little yellow dot. If we zoom in, this is an iron, okay? This is one atom of iron. Okay, so it's what allows the attraction of the oxygen to this. Okay. Now, iron is also an important cofactor in other enzymes. Okay, so we'll see in cellular respiration, aerobic respiration, we're going to see iron being very important in this transport chain. Okay, bringing things from one place to the other. Now, Let's define ionic bonds, covalent bonds, polar, nonpolar, all these fun bonds. This is very important for the classes to come. So let's talk first a bit about the chemistry of valence electrons. Okay, valence electrons are the electrons furthest from the nucleus and with the most energy, right? So the outermost orbitals of the atom. These electrons, they're super crucial in determining the chemistry of the atom and whatever influence it has on the atoms. So you can determine the valence by seeing the number of covalent bonds that an atom can form. Carbon can form 4, nitrogen can form 3, oxygen 2, hydrogen 1, right? The chemistry of these valence electrons, right, depends on which of these columns you're in, right? Group 8 is very content. It has a full shell and it's like, peace out, I'm fine. Then you have group 7 who's frustrated because all they need is one more darn electron and they'd be happy and have a full shell. So they're very reactive. And then group 1 is like, just take this one electron and it'll make me happy. Get rid of this electron, right? So that is what determines chemistry. Chemistry is all about, well, pretty much all chemistry is about these valences, okay? So we're going to talk about bonds, chemical bonds, not James Bond. And so to do this, right, let's just think of what bonds are. Bonds are attractions between two atoms. And here they write or molecules. I tend to not call it a bond because we have this image of what a bond is. I like to call it a interaction, right? Or a it's not as it's between two molecules, right? So we'll take a look at that together. So as I said in class, it's no magic stick. It's not like a legit tug of war between two of the atoms, right? It's really either sharing or giving away something. So let's start with covalent bonds. Oh, so this is split in two, by the way. There's intramolecular forces and intermolecular forces. Intramolecular forces are bonds between the atoms in the molecule. Okay, in the molecule. Inter is between molecules. That's what I was talking about, how it's not really, I see it more as a interaction between two molecules as opposed to a tight bond, okay? So covalent bonds, we're going to start with those. So what are covalent bonds? They are so strong, okay? A covalent bond is the sharing of a pair of valence electrons by two atoms. This is an intramolecular bond, as I said. So it's the strongest form of bond in organisms since they require continued close proximity of atoms to one another, right? So first, we have these, let's look at different types of covalent bonds. We have single covalent bonds where you're sharing one pair of the valence electrons, 2 electrons in total, right? It's a very long bond length because things are a bit further away, and it's the weakest out of these 3. Double covalent bonds, right? They're sharing 2 pairs of valence electrons. So you have 4 electrons in total, right? This is a kind of a medium bond length and an intermediate strength, okay? Triple covalent bonds, N for instance, right? 6 electrons in total, 3 pairs of valence electrons. Very short bond length because they're pulling each other so close, right? When you have these, the more bonds you have, the more tighter the pull is, right? And they're the hardest to break. It's like you have to break through 3 strings. Okay, now let's move on to, this is a very important concept for the rest of the year, polarity. nonpolar and polar covalent bonds. Okay? So here you have hydrogen and oxygen coming together to form water, right? What types of bonds are found in water? Well, between the hydrogen and the oxygen, right, there is a bond, right? And between the other hydrogen and the oxygen, there is another bond, right? So these are hydrogen and oxygen. One is really wanting to give away its electrons, the other doesn't. So this is going to be a covalent bond, right? These are covalent. It's between two nonmetals, right? What now? How many bonds are there? Yeah. So when hydrogen and oxygen comes together, right? How many bonds are there? There's two bonds that are formed. Each of them share two electrons, one pair, right? So it gets a bit confusing with the terminology, but don't get too stumbled on it. Now let's look at ionic bonds, okay? So before we do that, we'll talk about polarity, okay? Nonpolar and polar covalent. We're still in covalent. So what makes it covalent though? Like I didn't explain to you, but why is it called covalent and what does that entail, right? I like to think of these covalent bonds as a tug of war, okay? Polarity is the distribution of electrons. In a nonpolar covalent bond, the electron pairs are shared equally between the two atoms and are equally electronegative. In a polar bond, the electron pairs are not shared equally because one atom is more electronegative than the other atom. Unequal sharing of those electrons, they cause a partial positive we call dipole. You see that little delta sign, right? There's a positive dipole towards the oxygen and a negative one towards the hydrogen, okay? You have these little dipoles here. The more electronegative an atom, the more strongly it pulls the electrons towards itself. And the bond electrons will spend more time around the more electronegative atom. So this top one, when it's a nonpolar, right? It's as if the elements they're acting fair. They're putting 3 on each team. Then you get this big guy for polar molecules that's like, come here, and he just snags and you fall over, right? So that can be it. Let's say fluorine and fluorine would be a good example, or chlorine when it's paired with different things because it's highly electronegative, right? So the attraction that a bonding pair of electron feels for a particular nucleus depends on the number of protons in the nucleus, the distance from the nucleus, and the amount of screening. All you need to know is that electronegative pull, it comes from the periodic table's properties. And there's a trend. As you start in the very left lower corner of the periodic table, this is some of the least electronegative, the ones that pull the least to gain an electron, okay? And as you move up to the top right, fluorine is the one that wants an electron so badly. It's the one that wants it the most, okay? So it will fight with anyone to snag off an electron. It's highly electronegative. Electron, negative, it wants that negative charge, right? Okie dokie. Now we can move on to ionic bonds, right? So covalent means sharing of the bonds. They could be polar covalent, right? When there's a dipole, let's say a carbon and an oxygen, right? Oxygen will pull away more from the carbon. What else? Now we're going to look at ionic bonds, okay? So, first, before we do that, we'll talk about some ion stuff, okay? Ions, right? Atoms are electrically neutral, right? The number of protons and the number of electrons, right? That when they're equal, that makes them neutral. But they can gain or lose an electron and become an ion. That's what we call an ion. Positively charged ions are called cations, and they have a net positive charge with more protons than electrons, right? Negatively charged ions are called anions, net negative charge with more electrons than protons. So here's a list of a couple examples of cations and some of anions. You'll realize some of them are a bit longer than just one element. That's because those are polyatomic ions, more than one atomic ions. Okay, so this is a good way to remember it. Cations are positive, right? And here it says onions make you cry, so anions are negative. Onions. I get it now. That was a bad joke. So let's go through the ionic bonds. So they could be strong, but they could also be weak. Like it really depends on the conditions, okay? So an ionic bond occurs when an anion, a negative anion, right, strips the electrons away from the cation. So here chlorine is stripping away the electron from sodium. This forms an ionic salt, okay, which exists as a large lattice structure, ions of different charges stacking one on top of the next, okay, very close to themselves. Now think about it. Adding water to an ionic salt causes the dissociation of the ions, right? Since water starts to interact with different charged particles. Now I asked you in class, this was a very hard question, but why is it that the digestive system of animals must be adapted to break down covalent bonds? But you never heard of a NaClase or a KClase enzyme to break down those sugars? We do have lactase, right? Amylase. But why not for these, right? Well, it's very simple. It's that basically, remember that last point. Adding water to an ionic salt causes dissociation, right? In aqueous solutions, these ionic bonds will break apart, right? So, yeah, I think that was it for that. So basically, the covalent bonds do need something to break it apart, but not the ionic bonds, since they dissociate themselves to their simpler form. Now it says to find these other interactions. So we looked at all the two intramolecular forces. Now let's look between two molecules. What is going on between two molecules? So let's start with hydrogen bonds, right? These are very weak, but they are collectively small. So I pointed at them with the arrows, okay? When a hydrogen atom covalently bonded to one electronegative atom is attached to another electronegative atom, right? Usually an oxygen or a nitrogen of the other molecule, right? That is a hydrogen bond, okay? So when a hydrogen atom covalently bonded to one electronegative atom is also attracted to another one, okay? So look here, these water molecules, right? One of the hydrogens is very attracted to the negative oxygen of another molecule, right? So there's a little friction there. So no physical bonds are there. That's why we draw these dashed lines, right? Now, individually, hydrogen bonds are weak, but collectively, they're strong. And so this is why they are found holding DNA strands together, right? The strands must be stable, but at the same time, they must be pulled apart for transcription and replication to take place, right? So they kind of have to unzip all the time. So it's good that these are can be easily broken and easily put back together. So for another example, right, hydrogen bonds are also important in enzymatic activities, right? So let's take a look at these over here. So for example, so some enzymatic activities include substrates that bind and then unbind, right? So we'll see many examples when we get to those enzymes later on. One of the biggest areas of confusion for people is what a hydrogen bond is. It seemed a bit simple, but it's not, right? OH bonds, right, in water, OH at the top, that red and gray molecule, OH bonds are polar covalent bonds. Those are not hydrogen bonds. Yes, there's a hydrogen, okay, but it's not a hydrogen bond. It's a covalent bond, okay? This is an intramolecular bond. Look below, the O and the H bonds, right, are of the NH3, right, the blue molecule with the three gray things. Those it should say NH there. Those NH bonds are polar covalent as well. They're still intramolecular. Hydrogen bonds are between molecules. And remember, we put that dashed line. So between, let's say in this case, the hydrogen of the water and the nitrogen that has this dipole, right? So basically, we didn't take a look at it, but let me draw it. So when you have water, right, you have this oxygen over here, right? Then we're going to have a hydrogen, right? Okay, let's draw that here. And then we're going to have another hydrogen over here, right? So the net effect here is a dipole in this direction. Oxygen is going to attract the electrons. Same thing on this side, right? The net direction, right? These, if I decompose this into vectors, right? Like this and like this, right? They're going to cancel out on the x, but there'll be a net dipole of this molecule going this way, right? Going up. So oxygen is even more electronegative. It wants to go talk to other people and make these little attractions, right? Okay. So why is H2O the solvent of life, right? So a single molecule of water is kept together by polar covalent bonds between the oxygen and the hydrogen atoms, right? While the water you drink is made up of trillions of water molecules held together by weak hydrogen bonds. So that's what we just discussed. A single atom of water, molecule of water, will have these covalent bonds, but then in a glass of water, right, all these water molecules have these unstable little hydrogen bonds that are keeping it together. So when a spider, those water spiders, kind of float around on the water, that's why it creates this surface tension, because there's so many little forces amongst the water molecules, okay? Each hydrogen can form one hydrogen bond with another oxygen, and each oxygen have two hydrogen bonds with other hydrogens, okay? So the ability for water to form hydrogen bonds is what is really remarkable in this scenario, right? So, yeah, these covalent bonds are both polar and allow hydrogen binding between water molecules, right? And each of those hydrogens can form one H bond, okay? So that's what we just went through. Now these van der Waals interactions, right? These are still intermolecular forces, right? There's three of them. The first one is called dipole-dipole. These are much weaker than the hydrogen ones, all of these 3 van der Waals ones. So dipole-dipole interactions is attraction between 2 pairs of molecules that do not involve hydrogen. So look here. For example, CO bond, right? You have a slightly positive charge to C and a slightly negative charge to O. It's not big at all, right? If you go look at the electromagnetic table, they're not big at all. But it doesn't matter, right? There's no hydrogen, so we can't call it an H bond. So CO will tend to align with another CO bond so that they can, so that the slight negative oxygen of one is near the slight positive carbon of the other, right? Now, dipole-induced dipole interactions, right, is when a dipole from a polar bond causes a non-polar atom to bond or act as if it were polar. So it is inducing polarity, right? In what was originally a non-polar molecule, right? And this does not involve hydrogen. Okay, so look at this. We have isolated oxygen, non-polar. They're playing equal tug of war. The minute you bring this water molecule or another molecule close to it, right? Let's say there's this negative dipole for the oxygen, right? It'll be more negatively kind of have this dipole of negative charge. If you bring it closer and closer to that other oxygen, all of a sudden the oxygen goes, ooh, what's that over there, right? So then you you induce a dipole just by approaching it slowly close to it, right? That's induced. Now, London dispersion forces, right, are interactions between molecules with non-polar bonds, but still have a mild preference to interact, right? It's caused by the instantaneous dipoles, which occur due to chance, right? It's chance that when the electrons happen to be all more concentrated in one corner, right? So, for example, long carbon and hydrogen chains that tend to align with one another, that can explain this, right? These instantaneous dipoles. Okay, let's differentiate between hydrophobic and hydrophilic, okay? So, hydrophobic and hydrophilic, like dissolves like, hydro, water, philic, loving. So here, you have water on the left, right? And water is polar. And polar molecules is green and gray. One is polar, right? Which means it has a polarity. It has a certain dipole, right? It dissolves in water, right? Because they like to form hydrogen bonds with the water. So it's happy. So water is an excellent solvent. Water is polar and can dissolve any polar compound, anything that has a charge or a partial charge. Compounds that dissolve in water are considered hydrophilic, water-loving, right? Now, compounds that are non-polar do not dissolve in water, right? Because water is a polar substance, right? So these are termed hydrophobic, fearing of the water, okay? So we're going to see a lot of examples of this in our class, but let's take a quick look at the cell membrane's lipid bilayer. Okay, here is the lipid bilayer that encompasses the entire cell. Okay, you have these little lipids, right? So they have a funky little shape, right? And why do they form a bilayer? Well, I told you like will attract like, right? So the head, this is actually called an amphipathic lipid, right? Amphi means to change, right? So it has both a hydrophobic and hydrophilic property, right? The tails are nonpolar fatty acid chains, okay? And then the head is hydrophilic. It loves water, okay? It has a polarity. So when you're in the medium, these things will kind of flip on themselves, and you will form a layer where the glycerides will touch each other and stay together because they're happy, but also the heads will poke out into the cytosol or outside because it's very happy of that environment too. It's nice and polar, right? So the last little chem subject that we need to do is some acid and base stuff. So water, okay, is in a state of dynamic equilibrium. Okay, water molecules dissociate at the same rate they are being reformed. So you have two H2Os here, right? And though they can dissociate into hydronium, right, and hydroxide. Hydronium, we used to always write H+, but now we're in big boy chemistry, so we know that it's a hydronium. It makes sense. Look at those two H2Os. One of the H's needs to go to the other water molecule, right? The other one is a hydroxide. So for acids and bases, right, changes in the concentration of hydrogen and hydroxyl can affect the cell's chemistry, right? Concentrations of hydrogen and OH minus, right, are equal in pure water. It's pure, right? All right, so adding acids and bases modifies these concentrations of hydrogen and hydroxide. And so the pH scale is then used to describe whether a solution is acidic or whether it's basic. Okay, so these acidic solutions have pH values less than 7. Lemon, batteries, right? All of these things, tomatoes, right? Something a little oof, like it has a strong taste, right? Basic solutions have values greater than 7. Bleach, soaps, right? They're very slippery too. So this is very important. Most biological fluids have pH values in the range of 6 to 8. Okay, so that's like kind of the norm of where your pH should be, and if it's not, then that's really not good. So let's go again through some definitions. So acids, right? Any substance that increases the H plus concentration, right? So HCl comes into solution, hydrochloric acid comes into solution, and it right away gives away its hydrogen, says thank you for letting me in. H plus, right, gets released. Bases, on the other hand, is any substance that reduces the H plus concentration of a solution, right? So whatever can pick up H plus from the solution, okay? The pH of a solution is defined by the negative logarithm of H plus concentration, written as pH equals the negative log of the concentration of this proton, right, the H plus. Now, the concentration of this hydrogen, you can do the inverse, right? If you inverse a log, this is way back when mathematics, okay? But if you inverse this log, then to find the concentration of this H plus, you can just do 10 to the negative pH, right? It's a log scale. You're gonna jump up every time you do one step in the scale, right? You're gonna go up by tens, times 10. So for a neutral aqueous solution, right, the concentration of hydrogen is 10 to the negative 7. So the pH, well, what is it? It's going to be minus 7, right? So which will be 7. Okie doke. Let's keep going. So acids, right, that do not dissociate significantly in water are weak acids. All right? But we don't need to worry about that right now. So I'm going to designate an acid by HA. It's an acid, and I just want to show that it has the proton, it has the hydrogen. When an acid donates its proton, what are you left with? Actually, you're left with a conjugate base, what we call a conjugate base. You're left with a base because you got rid of that hydrogen, right? That's the conjugate base. And so the strength of an acid can be determined by its dissociation constant, and that's called the Ka. What does dissociation constant mean? Well, if an acid like hydrochloric acid dissociates very well, right, which it does, it can donate protons all the times, it can donate these hydrogens all the time, right? Then it's very strong and it dissociates a lot. So you'll be left with what? A lot of product, not a lot of the reactant. It'll use up a lot of all the reactant, right? So this Ka, let's see what the formula is, right? It's the dissociation of acid expressed in this reaction, right? So you're going to do the reactants on the top multiplied. Sorry, the products over the reactants concentrations, right? Whatever you have the most of in the end, right? Well, if it's reactants, it means that the acid didn't really react much. So the Ka, if you have a lot of the reactant HA, the bottom would be big, so it would be a very small Ka. A small Ka means this was a weak acid and didn't dissociate much, right? The other way around, if you got a lot of product, which is like the top numerator, right, then your Ka will be very big, and you're going to have a very strong acid. So this is exactly what it said. When Ka is less than 1, then the concentration of the reactants is greater than that of the project, is not significantly dissociated. Okay. The lesser the value, the weaker the acid, right? So similar to pH, the value of Ka can also be represented as a pKa, which is annoying. Just this isn't the most important thing for the course, honestly. It's just a lot of review. pKa, right, is specific to a substance, right, and tells you what a molecule will do at a specific pH. So the pKa is the pH value at which the chemical species will accept or donate a proton. So the lower the pKa, the stronger the acid. We don't need to go through all of this necessarily. Here they just do a little calculation using that formula that was given to you. Okay, so you can try that out on your own. Now, let's give the normal pH range of blood. So, internal pH of most living cells, okay, it must remain very close to the pH of 7. If not, it's not good. How does it do that if we're exposed to all these acids and bases all the time, right? Buffers are the answer. Buffers will minimize changes in the concentration of hydrogen, right? H plus, the proton, and OH minus, okay? So buffers contain a weak acid and a corresponding base, which combine reversibly with H plus ions, all right? So basically, let's look over here. The pH is a mathematical representation of the concentration of free H plus, right? So these buffers are minimizing the change of hydrogen. If we add an acid, right, we're adding H plus, right? H plus will bind to this bicarbonate, this on the right, HCO3 minus, right? That's bicarbonate. Therefore, the concentration of free H plus will change minimally. If you add a base, right, let's say you add OH minus, right, then the H2CO3, right, this acid, right, will donate, carboxylic acid, will donate a H plus, right, and therefore the concentration of free H plus will change very minimally. So whether there's an acid or a base, it's a buffer. It's like, whoa, calm down, you, and it'll give a bit more of the opposite so that things don't hit the fan, right? Okay. let's start the fun stuff now. Let's try to differentiate between organic and inorganic molecules, okay? So, organic molecules, right, have carbon backbones. I don't know if you see very well over here, right? But we're gonna, if you didn't take organic chemistry, that's fine, but we're gonna talk a lot about these this year. So it's good that we're gonna start getting ready to see this org stuff. So for example, you'll see here that um, these molecules all have this carbon backbone. Oop, let me just grab my pencil. So over here, right, you have this carbon backbone that's all linked together in a zigzag formation, right? Look at this glucose 2. There's all these carbons that are at every corner, right? The way that organic chemists write it is just lines. They just represent lines by the carbon, and it's assumed that we know that those are carbons, okay? Because we don't want to draw this thing every single time. We'd rather just draw these lines, okay? So it as opposed, so there's exceptions, of course, to this rule. These are the carbon molecules that are not organic, okay? So simple oxides, blah blah blah. You don't need to memorize them. I just put them there for your information. Now, inorganic molecules, well, they're not bound to the carbons, right? Does that make sense? So basically, organic molecules, they have a carbon backbone. Inorganic phosphates are an example of an inorganic molecule since it is not bound to carbon. Okay? Now, question. Why is carbon so important, right? Carbon is actually extremely versatile, okay? Carbon can form 4 covalent bonds, which allows it to form chains, branches, and rings, okay? So carbon can form double or triple bonds as well, and can bond to many different elements. Most often, it'll go bond to oxygen or nitrogen or hydrogen, okay? So very few other elements are capable of such variety. Carbons allow the formation of very diverse range of molecules, which we'll discuss in the next few sections, okay? And so this tetravalent nature, being able to make those 4 covalent bonds is very important. Okie doke. So, the 4 main classes of organic molecules. Look at these carbohydrates. You see these rings everywhere that you see a connection between lines and that there's no letter written, that's a carbon. Every time you see a C, that's a carbon, right? So you see all of these carbons, hydrogens, oxygens, they form these rings. That's very important for carbohydrates. We'll see this later on. We have these carbohydrate rings, right? This is one monomer of a carbohydrate. This is actually lactose, okay? Proteins, right? They have this carbon backbone, boom, 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 right? And then they have these little groups at the end, right? And oxygen sticking out here and there, right? So that's very important. Let's look at nucleic acids, right? Oh my God, these are complicated. We have a lot more going on here, but we have some phosphoruses. We have these rings again. So we'll take a look at those in due time, all right? And then lastly, we have these lipids, okay? Lipids, right, are these long carbon chains, okay, that never end and go on and on and have these little double bonds. So this is a fatty acid. Now, I don't know if all of you took organic chemistry, but I want to bring up an important point, okay? When you have these, you'll see over here that you have all of these carbons, right? If it is not specified, let's say here in the lipid, right? In the lipid, we don't see it, but this little junction is a carbon, right? All these junctions, this junction's a carbon, this junction's a carbon, this junction's a carbon, right? So, and carbon, remember, we said always bonds in pairs of, it bonds to 4 other things. So actually, this carbon here, let's say, right, it's bonded to this carbon here and here, right? But that's just 2 bonds. That's 1 bond here and 1 bond here. So what else is there? Well, organic chemists are very lazy and they don't want to always draw hydrogens. So actually, over here, there'll be 2 hydrogen bonds as well. If there's nothing else there, it means there's a hydrogen. So this carbon, let's say that's right over here, right? There's a carbon it's bound to here and here, right? That means over here as well, there's a hydrogen, right? Because carbon always needs to make those 4 bonds. Let's think of this over here, okay? We have a double bond and on the other side a single. So this is 1 bond to a carbon. This is 2 bonds. And this is double bond, so 3 bonds. So actually over here, there's just 1 little hydrogen, okay? Over here, you'll see that this carbon here is bound to 1 oxygen. The oxygen is then bound to the H. When it's conjoined like that, it means there's an order. It's not like the carbon is bound to oxygen and separately H. It means that it's bound to O, and then O is bound to H, okay? So this carbon is bound to O, this carbon is bound to C, and this carbon is double bonded to O here. That's 4 different groups. That's 4 different bonds. So this carbon is happy. It actually doesn't even have a hydrogen partner. Okay? So, all right. Let's keep going. So the next thing we want to do is understand what isomers are and differentiate between structural, geometric, stereoisomers, okay? All those fun things. First, we're going to look at the distinctive properties of these organic molecules. There's 2 important parts to these organic molecules. The first thing is the carbon skeleton. Remember I told you this is the skeleton of all these little carbons that are lined up in a row, right? This is the backbone. This is why carbon is so prevalent, right? So let's take a look at this carbon skeleton. The architecture of organic molecules varies, right? The skeleton varies in length, in branching, in double bonds, right? So let's practice drawing these organic molecules. This is C and another C, right? So if I were a chemist and I were to be annoying and just draw this, right? I would probably draw something like this, just a line. Because you have 1 carbon here, right? And it always needs 4 bonds. It's bound to this other carbon that's over here, right? But this carbon will have 3 other hydrogens here that I'm not going to draw. Remember, chemists are lazy to draw the hydrogens. So if I were a chemist and I were to draw this molecule of ethane, eth means 2, okay? So I would just draw a single line. Propane, prop is always for 3, right? Propane has 1, 2, 3 carbons. So I can go 1 to carbon 2 to carbon 3. We always do those zigzags, right? And again, I'm a chemist, so I'm lazy, but I'm not going to draw in that there's 3 hydrogens here, right? And I'm not going to draw in that there's 3 hydrogens here. And I'm not going to draw in that there's 3 hydrogens here because I'm a chemist and I'm lazy. But know that they're there, okay? And then lastly, let's go look at some other funkier things. Here you have 1 butene, okay? So 1, 2, 3, 4. So this is going to be 4 of these bonds, right? So 1, 2, okay. So butene, right? You're going to have a carbon here, 1. It's going to go up 2, 3, 4. Now, This carbon has 4 bonds. This carbon has 4 bonds, right? 1, 2, 3, 4. This carbon, however, right, has 1, 2, 3. Actually, it's butene. This here you don't see it as well in the PowerPoint, but there's a double bond here. So actually, we'll go draw a double bond here, okay? And then this carbon is happy because it only has 2 H's, right? It has 2 H's, and so does this one. This one has 2 H's as well. Sorry, this one only has 1 because it's bound to the other carbon, right? This carbon here is bound to 1 carbon here, has 1 bond, and it has 2 bonds here. So it's just going to have 1 other hydrogen, okay? This might be all review, or it might be the complete opposite and super new, and you're like, what is this? Don't worry about it. We're going to see these things over and over. It's organic chem. Not to worry. 2-butene means that the double bond is in the second position. You don't need to memorize all these things. I'm just showing you a bit what it looks like when we draw the stick version, right? So 1, 2, 3, 4 of these. So this carbon has 4 bonds, so it's okay. Then this carbon does not, right? That means the double bond is going to be here. We don't see the double bond that well where they drew it, but it is there. You should be able to see it in your manual if you look, okay? So 1, 2, 3, 4. 1, 2, 3, 4. There we go. That's 2-butene. Okay. Butane, just normal. This is a normal butane without a double bond. This is butene with that double bond, right? So this would just be 1, 2, 3, 4. Easy peasy. This one's a bit weird. This is 2-methylpropane, right? So we're going to start 1, 2, 3. We have a chain of 3. 1, 2, 3. Oop. Undo that. So 1, 2, 3. And then in the middle carbon, we have a little methyl group. We have this methyl that is coming out, okay? So that's how that one would look like. This one is a cyclohexane, right? So it's going to look like this. That's all we need to draw. And realize that each of these carbons only attach to 2 hydrogens because it's happy with its 2 carbon partners, right? Benzene is a little bit more of a crazy one, even chemically. But we have this ring, except that every second part of the ring, there is a bond, right? So this, and oops, I didn't even draw this properly. Anyway, it has a double bond every second. So look, this carbon here, let's say, it's only bound to 1, and then it has 2 bonds, so it only needs 1 hydrogen. That's what's special about benzene, okay? Alrighty. Let's understand some more things about isomers, okay? So here, isomers are just compounds with the same molecular formula, okay? But different structures and properties. Let's quickly look at structural isomers, okay? They have different covalent arrangements, all right? So pentane, right? And you'll see pentane is C with 3 H's followed by C with 2 by C with 2 by C with 2 by C with 2, right? So let's write out the molecular formula for pentane. It's going to be 1, 2, 3, 4, 5. So C5, H, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. H12. Okay? This one here, 2-methylbutane, is the same thing, C5H12, but what? It's in a different arrangement. This one would look like 1, 2, 3, 4, 5. This one would look like 1, 2, 3, 4 with a branch over here, right? So that's why they say it's 2-methylbutane, because the methyl is on the second carbon, okay? Again, I'm going into a lot of chemistry, but it's just like so that you're comfortable when you see these molecules. You'll just need to know a bit of organic chemistry to get biology, okay? So these are isomers. Why? Because these two compounds have similar molecular formula, the same molecular formula, but different structures and properties, right? So these structural isomers specifically, they have different covalent arrangements. Stereoisomers, okay, have the same sequence of bonded atoms. So actually, unlike the structural atoms, they actually follow the right order. Look, you have this cis isomer and trans isomer, right? They are the same order. The only difference is in its 3D orientation. Okay? Cis isomers have, let's say, the two X's there on the same side. A trans isomer is going to be, those X's will be on separate sides. Okay? We're actually going to see this with fatty acids. We're going to see cis and trans fatty acids. You've probably heard of trans fats, right? Oh, so bad. So we're gonna learn about them and what does it mean? Why is it actually those different saturations change things? Okay. Enantiomers are mirror images of each other. Okay. They exist when carbon is attached to 4 different things. Okay. So enantiomers are basically you can orient these carbons in different ways. So they're mirror images, right? So some of them will be at the bottom of it will be hydrogen, methane, NH3, right? And on the other one, it's flipped. It's the mirror image. So it looks the same, but in 3D space, this is extremely important to know which methane is pointing straight at me, right? In this case, it would be which NH3 is pointing to the right. Because if there's a buddy on the left, maybe it's good that it's reacting with something to the left or having an intermolecular force. But maybe it's good that it's far away from it, right? So all these things we'll take a look at. So let's recap isomers. You have these structural isomers where you have basically just a change in the sequence, but it's the same molecular formula. Then we have stereoisomers, okay, which is basically that you're changing the 3D shape. For diastereomers, you have these cis and trans isomers, right? So cis would be the first one because the methyls are on the same side in space, in 3D space, and trans for the second one because they're on opposite sides. And lastly, the enantiomers, right? These are chiral, what we call chiral centers. We're going to look at that, and that's going to be our assignment in class. So these chiral centers, right, are bonded 1, 2, 3 to 4 different atoms, right? And we're going to take a look at those soon. So I'm going to break them down each again separately so that we really understand, okay? So structural isomers, or they call them constitutional, have different physical structures, right? They're in different orders, but they have the same molecular formula, okay? Isobutane or N-butane is the same. Cis and trans isomers are different orientations across a carbon-carbon double bond. Cis is on the same side and trans is on opposite sides. This is where we need to practice some stuff. Stereoisomers look structurally identical, okay? They have the same covalent relationship, but they have different stereochemistry, right? Their spatial relationship is different. So when carbon has 4 different groups attached to it, only carbons, okay? So we call it a chiral center or a stereocenter. Carbon needs to be carbon, and it needs to have 4 different groups. We call those chirality centers, chiral centers or stereocenters, okay? So it's going to be very important. Stereoisomers need at least one of these chiral centers, but we're just going to learn how to identify them. So look here. This carbon here, right? It's attached to a methyl, a CH3. It's attached to this other thing that I can't see, a hydroxyl, OH, and a hydrogen, okay? Over here, right, we have this molecule, and it's, you also see that it's attached to 3 different things. So this carbon, we say it is a chiral center. Chiral center is a carbon atom bonded to the 4 different substituents. Let me give you another example. What about this? CH, OH, a hydroxyl. We're going to have over here a sulfur, and here we'll have another hydrogen. This is not a chiral carbon, okay? Why? Because we have one hydrogen here and another hydrogen over here. That's not good. We need different substituents to be chiral, okay? Alrighty. So, a chiral center, right, or a stereocenter is a carbon atom bonded to 4 different substituents, right? So, let's look at this carbon first. Is this carbon going to be a chiral center? It's bonded to a hydrogen. It's double bonded to an oxygen. That double bond should right away tell us no. And then it's bonded to this other carbon. Well, actually, I'll take that back. It's bonded to all of this, right? So, what's going on here is that there's not 4 different types of groups, right? You only have 1, 2, and 3 groups. It's not like you have another hydrogen sticking out here. So this chiral center, this is not a chiral center. There's not 4 other atoms that carbon is bonded to, right? Okie doke. Let's keep going. This carbon over here, though, right, is bonded to this group, right? And then it's also bonded to this other hydroxyl group, and it's bonded to whatever all of this is, right? And there's no double bonds, so that means there's actually an additional hydrogen sticking out on the side over here. Okay? So this entire molecule, right? This point over here is a chiral center. All right? Now, for this molecule here, right? This carbon is connected to this group here, right? This carbon is connected to this different group over here. This is a hydrogen, and this is an OH. These are indeed, this carbon is a chiral center. It has 3 different groups. Let's go see the next one though, right? So this carbon, right, has a methyl group. It has this other group over here, right? And it has this bromine group. And it has this other thing. So it is a chiral center. Let's go see what is not a chiral center, okay? What is not a chiral center is this carbon here. Why? Because this is bound to this one group. 1, 2, we're doing good so far. 3, we're doing good. And there's the next one, so that should be good. No. Why is it not good? Because these are 2 repeated groups. CH3 is 2 of this. There's 2 methyls, right? There's 2 of these CH3s, which means you're not allowed. A stereoisomer cannot have 2 same groups, right? All right. We were talking about enantiomers before, right? So L-glucose versus D, right? You just switch it around the sides that it's on. This is not so important. It's diastereomers. These are not mirror images. You need to have a stereocenter for that, though. Let's not focus too much on it. But all I would say is to remember, right, that structure and function of biomolecules are inseparable. Thalidomide over here, right, the R isomer, which is a certain, when the chiral center is turned one certain way, that R isomer reduces morning sickness in pregnant women. The S isomer is not known to is now known to cause birth defects. So that's crazy that just one turn of the molecule could mess up everything. The drug was prescribed in 1960s and 1970s, and thousands of birth defects occurred. It's very sad. Okay, now remember we did the first part. That was all about the carbon skeleton. Imagine. But now there's even cooler parts. We didn't even talk about the molecular components attached to these organic molecules. We talked all about that carbon chain, but we didn't talk about these little thingy-majiggies that are all sticking out at the ends, okay? We're not interested in the hydrogens. There's all these hydrogens everywhere, but we're not that interested in them. We're interested in these functional groups. Functional groups are components of organic molecules that are involved in chemical reactions, okay? So the number and the arrangement of functional groups give molecules unique properties, all right? So here we have estradiol and testosterone, which differ in only 2 functional groups. Look at this. Here in estradiol, we have a hydroxyl group. And here we have no CH3, no methyl group, right? That's the only difference between testosterone and estradiol, right? Which is what's important for the development of males versus females, right? Like, that's crazy that just one of these functional groups can change it on this steroid, okay? So here are the 7 functional groups we'll look at. I would like you to get familiar with these. This is what's going to be a bit on the quiz, okay? So hydroxyl group, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and methyl. You'll get the hang of it very quickly because phosphate, oh, that's a P, right? Methyl, well, we talked about methyl a lot. It's that CH3. Sulfhydryl, there should be a sulfur somewhere. Amino is like amino acid. There should be a nitrogen, right? Carboxyl and carbonyl will be the hardest to differentiate, okay? But then hydroxyl is OH, right? Hydroxyl, OH. So these aren't actually too bad to learn. Okay, let's start one by one. Hydroxyl groups. When you have this OH appendage that's sticking out of a molecule, boom, you have a hydroxyl group. Look at this here. You have your little chain, and then all of a sudden you have a hydroxide, right? let me draw it out for you. Here you have a carbon, another carbon, and then here you're going to have this OH, right? I drew my first carbon, my second carbon, and then my OH. And remember, I'm not showing them because I'm a lazy chemist, but there's hydrogens here. There's 2 hydrogens here because there's 2 other bonds there. And here there's 3 hydrogens, right? But we don't draw them. These hydroxyl, right? Ethanol. This molecule here is in an amazing little glass of wine called ethanol, right? So it's the alcohol that's present in alcoholic beverages, okay? It is polar due to electronegative oxygen, okay? Look at this OH. Remember the OH? This is why we talked about bonds, right? OH, right? The O is pulling the electrons from the hydrogen, right? So it's polar due to electronegative oxygen and forms hydrogen bonds with water. So if you have this ethanol, right, like this OH, it'll form some bonds with water, right? Helping dissolve compounds such as sugars, right? The compound's name is called alcohols. You have different alcohols, propanol, any OL is an alcohol. Carbonyl group, okay? This is when you have a C double bonded to O. Okay, whenever you have that, that's a carbonyl. This one's a bit trickier, okay? But look over here. Here you have a CO. I see a CO over here, so that's a carbonyl, right? So when it's, you don't need to memorize this. I don't want you to memorize this, but when it's in the, you can call it the carbonyl group and you'll get all your points. Like, don't worry about it. Now, it's just interesting to know that ketones, right? If the carbonyl group is inside of the molecule, like in the first case, that's called a ketone. Whereas if it's on the very end, it's an aldehyde, right? Formaldehyde, all those things, right? You don't need to memorize that at all. Please don't. It's just for fun, but just call this the carbonyl group, right? And whenever you see that C double bond, except, this is very important, except when you have this OH. When you have C double bond O and there's an OH and it's hanging off of a molecule, look how it's just the end, right? Like this. Then boom, that you do not call a carbonyl. You will get some points taken off, okay? This is a carboxyl group. It's very, very different, okay? This is actually acetic acid, which gives vinegar the sour taste. And when it gets in its ionized form, right, it gives away a H. It's a very weak acid, but when it gives away its H, right, then it becomes COOH, right? So it's a carboxylate ion, right? This is found in cells. And it acts as an acid and can donate hydrogens because the covalent bond between oxygen and hydrogen is so polar, right? Now, so we call this carboxylic acid. So carboxylic acid, right? Or here they write organic acid. I've never heard of that. Yes. So again, don't memorize the functions and all that. I just want you to recognize the groups in a molecule. So this is an amino group, right? NH2, remember amino acid, there's nitrogen at one end. So an amino group, right, NH2, you have this glycine, for instance, which is an amino acid, right? The building blocks of a protein. And note its carboxyl group, right? It has a carboxyl group on one end, and it has this amino group on the other, right? That's the definition of a amino acid, okay? you're going to have one carboxyl on one end and one amino group on the other for amino acids. Now, when it ionizes, right, this amino can be found in its ion form, right? So here it says it acts as a base and can pick up hydrogen from surrounding solution, right? And then you have the other end, which is your carboxyl, which can actually go and donate a hydrogen. So this is kind of like that dual buffer kind of situation, right? This sulfhydryl, right? It's SH or HS, right? It's you see it like sticking out places. Cysteine. It says cysteine, a sulfur. So this is an amino acid too. How do we know it's an amino acid? Well, here I see a carboxyl group, and here I see an amino group, right? So those are both those are like the two important things to pick up. Okay. So just a little fun fact: cross-linking of cysteines in hair proteins maintains the curliness or straightness of hair. Okay. Straight hair can be permanently curled by shaping it around curlers, right? And then breaking and reforming the cross-link bonds. So that's how perm is done. Phosphates, okay? Here are our phosphate groups, okay? So P and all these O's around. Notice it doesn't follow the octet rule. It's an exception to that octet rule, okay? So here you have glycerol on the right. It's glycerol phosphate, which takes part in many important chemical reactions in the cells. Okay, so we'll see them later on. Anything with phosphate should ring your bell for that. And this last, I think this is the last one, the methyl group, right? This methyl is just a little is one little CH3 popping out. Okay? When it's linked in the chain, that's not a group. This is the chain. But if there's just one sticking out here, then we consider this to be a methyl group, and that's one of them that we'll write down. Okay? Alrighty. So, this is the summary, okay? And this is what I want you to kind of retain, alright? So, notice R is just continuation, whatever continuation of the chain, okay? All these little R's and R primes is just some kind of continuation of this backbone. Okay, now the fun stuff, okay? So what we're going to do is we're going to name the functional groups that we see in this molecule. This is going to be your quiz. Literally, not these molecules, but I'm going to give you some other molecules, and you're going to name the functional groups that are found, okay? So, first thing, what do we see? You can pause the video. I put the little table there for you too, since you're not familiar with them yet. I want you to go and circle all of the ones that you see. I'll do it with you this time, but maybe pause your video and try to do it yourself first. Give it any try, right? So, all right. So this, I remember this, OH, hydroxyl, right? Hydroxyl, okay. I see another one of you here sticking out, right? And then, what else do we see? So, sorry, I jumped a few steps, but where is the carbon backbone? Well, there's a carbon attached to a carbon attached to a carbon, right? This is kind of the backbone over here, right? And then from there, do you see any groups, right? So, now, I saw a hydroxyl here, but you know what? I see over here I see an O double bond. So I said it was a hydroxyl and a, let's see, carbonyl, but I'm super wrong, because look, remember I told you not to mistaken this the carboxyl, right? Carboxyl, carbonyl, they're very different. The carboxyl is whenever you have this double bonded O and the OH at the same time. So I'm going to erase my answer I put here and change it. I'm going to say that this over here is my carboxyl group. Carboxyl. Okay? Next, this over here, NHH, right? N2H was my amino group, right? Amino. I see it right over here, right? My amino group. Could also write it like this, NH2. Okay. What else? That's pretty much it, honestly. Now, if this was confusing for you, okay, what I would suggest you do is draw out the molecule in a way that makes sense to you. It's a bit twisted and annoying here, but if I drew it out like this, C connected, I'm going to start at this end, okay? C connected to O, connected to H, right? And then double bond up here I'm going to put, and then connect to C, just so that everything's in a straight line. And then there's an H, right? Notice nitrogen only needs 3 bonds. That's all it needs. And then here there's a C, and there's an H, and there's another H, right? And then here there's an OH, right? This helps you to see it in a little bit more visualized way, a stretched out way, as opposed to all the folded chains over here. But as I said, my tip is find your carbon backbone and then work from there. What's coming off of that carbon backbone, okay? Alrighty, let's go on to the next thing. Ooh, how many chiral centers are there in this molecule? Okie dokie. We're going to locate our carbons first. Carbon number 3. So carbon number 1 is a chiral center. What's the definition of chiral center? It's carbons that have 4 different, right, differing groups. So, what do I see here? I see an OH, I see an oxygen, double bonded, which means it's only going to have 3. This counts as like another bond, right? So it's only going to have 1, 2, 3, and this 4th one. O is double bonded. It's not going to have a hydrogen. If this was a single bond, then that would mean that there would be an extra H sticking out, right? Which would mean we would count it as a chiral center. But in this case, right, nope, this is not a chiral center because there's this double bonded O. Let's look at this next one here, this C here, okay? Here, for this C, we have this group here. We have a hydrogen. We have an amino group. And we have this other big thing here. These are all different groups, okay? They're all separate groups. So, what does that mean? That means that this is a chiral center. Last but not least, here we have an OH. For this carbon, we're going to assess if it's a chiral center. Here we're going to have this OH, right? Here we're going to have this hydrogen. Here we're going to have this big appendage of a group. And here we're going to have a hydrogen. Oh, it looks like there's 4 groups. But look, there's 2 of the same groups, hydrogens. That's bad. That means it's not a chiral center, okay? So all this to say that this is not a chiral center because the hydrogens, they were repeating, right? So I'm going to go and I'm going to say, nope, not that. This is not a chiral center, okay? Let me get rid of that because that's not a carbon. Okie dokie. Last one, okay? So for this one, right? Oof, there's a lot going on. Let's name some groups. Well, I see a methane here, right? CH3, methyl group, right? Sorry, not methane, methyl group, methyl. Okay, what else do I see? I see a OH, another OH, okay. Ilala. I see a carbonyl, another carbonyl, and they're not special ones. Remember, I'm always looking out for this in case it's a carboxyl, but it's not in this case, okay? So what am I going to do next? I'm going to go, ooh, I see another carbonyl here that's attached to the amino. I'm not interested in that. And then I have an NH2, right? An NH2 is that amino, right? Okay, so I think I'm doing good so far. Over here, I'm going to have a nitrogen with 2 methyls coming out. It's CH3, 2. So this is going to be 1 methyl coming out here and another one coming out here. So I count those as 2 other methyl groups also, right? On the quiz, I'm just going to ask you what are the groups there. So you don't need to necessarily count how many there are. If you would like to circle them, I'm not going to grade you on it, but I can then guide you and see if you missed any, okay? So maybe do put them there. Alrighty. How many stereocenters are there? Ugh, this is going to be a disaster. Well, you know what? Let's just label all our carbons. I know there's a carbon here, a carbon here. Every junction, right? Carbon, carbon, carbon. We're literally squishing organic chemistry into like one day, which is great because biology is the best. So I'm going to label all my carbons. Carbon, 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 carbon. Oh, sorry, I was doing the stereocenter. Yeah, exactly. Alrighty. Let's start. Okay. So, this carbon here, I see a double bond right away. I know there's not going to be 4. Whenever you see a carbon and it's linked to a double bond, get rid of it. There can't be 4. It can only make 4 bonds, and that double bond is taking up 2 of those, right? So double bond, here I see a double bond linking one. Here I see a double bond linking one. Here I see a double bond linking one. Here I see it, and here. So look how much I got rid of, right? Now, here I also see a double bond linking it. Get rid of it, right? Here I do not see a double bond, and I say this is our first one. You have this group, like all that stuff. This group, you have this methyl, and you have this hydroxyl, right? So that's looking good to me. That's looking really good. So we gotta go check it out. We have this group here, right? Like it continue. It's not just that group. It's like all of this group, right? Like it's considered like all of that. So you have that group there. You have this group here, right? And then you have this group here. And then that's just 3 bonds. Oh, yeah, there's a hydrogen, right? So they're all different. I would consider that to be a chiral center, right? I think that is a chiral center. So this one here, boom. I'm going to say, yep, this is a chiral center. So I'm going to get, I'm going to write 2, right? And I'm just going to put my little hydrogen so that I know that there's a hydrogen there. Up here, right, you're going to have 2 H's coming out because there's only 2 other bonds. So this is not a chiral center. You can't repeat atoms, right? So this is not. This one here looks like a chiral center to me because we have 1, 2, 3, 4 different groups. So I would say this is our third one. And then this one here has a nitrogen. It has this group. It has that group, right? So I think this is a fourth one, right? Fourth chiral center. What else do we got? Fourth. Then over here, there's a double bond. Remember, we can't touch those. Double bonds mean that you're wasting one of those 4 bonds already. Double bond here, right? Double bond here. Oh, here we have a double bond. Here we have a double bond. This one, we can take a look at. That one looks good to me, right? Over here we have one group. Over here we have another different group, and we have another one, and we have an OH, right? Nothing's repeating here. So this one I would consider our very last one. So I think there's 5 in total. There's 5 of these, okay? All right. So that was a review of all that gross organic stuff and some other chemistry, all right? So I know next class is coming up quick, and you probably just skimmed through this and got a bit out of it. But make sure that eventually, maybe this weekend, you have the time to review this video properly, right? Review lecture 1, this organic chemistry section, right? Okay, so, this is actually the slide for next class. So on Thursday, tomorrow, we're gonna review lecture 1, right? So I'm gonna do a bit of the organic chemistry. Then we're gonna play the Name That Group game. It's the quiz, okay? So your grade is I think it's 0.5% of your grade, okay? And basically, I'm gonna show you 4 molecules, okay? And you'll need to name the chemical groups like we just did, exactly like that. You'll get 2 marks per molecule, and then name how many stereocenters there are, okay? Kind of like we did. They'll be a bit tough, so I'll see how much time we get through and see what we're going to do about that, okay? And then we're going to start lecture 2, which is on carbohydrates, okay? If somehow you miraculously do all of this video, you read the chapter 1 that was in the notes, and you have nothing to do, and you feel ready, then you can go ahead and pre-read. I always suggest to pre-read before class, right? So if you have time, go read the notes, the lecture notes for pages 8 to 13, which is on the carbohydrates, which we're going to start next class, okay? So that is it for me. And I'll see you on class Thursday. Take it easy, everyone.